Hello everyone and welcome to this edition of TalentsTeach. And today we have a very special guest with us because the topic is also very special in a way. The topic is on stem cells in ophthalmology, a very elusive, a very difficult to understand topic. And I think I've chosen the right person to talk on this topic. She's Dr. Yogapriya. She's a senior research fellow from Aravind Medical Research Foundation in Madurai. She has just finished her PhD in biomedical sciences, and I think she will beautifully illustrate us what stem cells are all about when it comes to ophthalmology. So over to you, Yogapriya.
Yeah. So thank you, Dr. Pranesh. So that was a very warm introduction. So as Dr. Pranesh told, today we'll be looking into stem cells. So the major emphasis will be on stem cells in ophthalmology. So moving into the presentation, this will be the content of the presentation. First, we'll touch upon the introduction, the basics, the properties of stem cells. Then we'll get into the types of stem cells and the role of stem cells with aging and disease pathogenesis, and stem cells in ophthalmology, our main topic. And then comes the adult ocular stem cells, location and function in each part of the eye, and then the therapy associated with it, and very importantly, the challenges in bringing the ocular degenerative therapy to patients and my references. So now let's move on to the basics, introduction on stem cells. So let me take it from the very basics. So stem cells, they are undifferentiated, or it's the most primitive cell, I should say. When I use the word undifferentiated, it means it has no identity of its own, which means, so we address a cell as a neuron, a hepatocyte, or a muscle cell, but when you say a stem cell, they don't have any of these identities, but they have a greater potential to divide into any cell of or any cell of a particular lineage. So it depends upon the type of the stem cell. So what is the primary function of the stem cell? So the primary function is to maintain the tissue homeostasis. So to keep all function running normally in a human system, it's been governed by the stem cells, which is present in various parts of the human body. So this is the ultimate aim of a stem cell. So there are certain properties of stem cells by which you can define them. The first property is quiescence. So the term quiescence, it means dormant. Stem cells, they are not proliferative. They are not, I should not use the word not proliferative. They are not actively dividing. So they don't actively divide, they remain dormant, but they have a great potential to divide upon a need. So whenever there is a mechanical damage in the body, whenever there is a disease pathogenesis that's going on, so whenever there is a repair mechanism that has to be started, a stem cell comes into play. So till then, they are dormant in their niche or microenvironment. So next comes self-renewal. So the term self-renewal, it means whenever a stem cell receives a signal to divide, they divide into two cells. One is a stem cell and the other cell is a progenitor cell, which move towards a specific lineage where the repair has to be taking place. So self-renewal, to be simple, it is a copy of making a stem cell as well as a differentiated cell. The main purpose of making its own copy is to maintain the stem cell population. It is there are two types of division, a symmetric division and asymmetric division. The one that I have shown in the image here is the asymmetric division, where a stem cell divides to give one stem cell and the other progenitor cell of a particular lineage. So it maintains its population by dividing into one stem cell and other differentiated cell. And now coming to the potency to differentiate. So whenever there is a signal could be an interleukin, it could be a protein from the microenvironment, which actually stimulates or triggers a stem cell to differentiate, divide and differentiate. It is known as the potency. So a stem cell, it can divide into any cell or when I talk about the types, I will explain this in detail. So it has the potency to divide into two or more cell types. So let us have it two or more here. So a stem cell, it could become a blood cell, or it could become a chondrocyte, it could become a beta cell, neural cell, or it could be a myogenic one, muscle cell, etc. And now, moving into quiescent versus activated stem cells. So the reason behind projecting this slide is like how we researchers look into stem cells. So based on what properties we define a stem cell, a quiescent stem cell, or it is an activated stem cell. So in my lab particularly, we define a stem cell based on size. When a stem cell is one which is actually smaller in size, it has a high NC ratio. NC ratio is nucleus to cytoplasmic ratio. A stem cell is a small cell. It has a larger nucleus and it has a very thin rim of cytoplasm. Because it is dormant, the metabolism is so low and histone methylation is much less. So these are all some cellular properties based on which we define a dormant stem cell. So whenever a stem cell receives a signal from the microenvironment to divide, the cell size becomes bigger because the cellular machinery is becoming active to divide. So the cell size becomes bigger. So it's just opposite to what a quiescent stem cell looks like. So these are certain cellular characteristics or physiological characteristics we look for in a dormant stem cell and an activated stem cell. And moving into the type of stem cells, so there are mainly 3 types of stem cells. So we'll spend some time here on this slide. Embryonic stem cells. So talking about their potency, meaning to differentiate, they are either totipotent or pluripotent. So embryonic stem cells, they are isolated from the blastocyst stage of embryo. So the first few divisions of the inner cell mass, they're totipotent, which means they can give rise to any cell of the body, including the extra embryonic structures like placenta. And after the 2 or 3 divisions, they become pluripotent. They can become any cell of the body, but not the extra embryonic structures like placenta. So as given in the image here, from the inner cell mass, it can be isolated and they have a great potential to become any cell of the human body. And now coming to adult or tissue resident stem cells or somatic stem cells. So these stem cells, the potency is much limited. It is not as embryonic stem cells. They're either unipotent or multipotent. So these stem cells can differentiate into like 2 or more cell types. They don't have a potency like embryonic stem cells. So the stem cells of the blood, the hematopoietic system, so they can differentiate into RBCs, WBCs, monocytes, leukocytes, etc., and other blood cells, but they cannot give rise to any cell other than a blood cell. So this is what it means. The potency is much less than an embryonic stem cell. And sometimes they divide into and then they move towards only one particular lineage. So they cannot differentiate into any other cell except a specific lineage. So that we call it as unipotent. And the other case that I was talking about is multipotent. And coming to the induced pluripotent stem cells, and this is one fascinating field, and a lot of research is being carried out using induced pluripotent stem cells because it's very easy to work with iPSCs, induced pluripotent stem cells, as well as embryonic stem cells because of their potency. And in case of adult stem cells, it's a little challenging working with adult stem cells because I'm one person who's working with adult stem cells because of the number. Because adult stem cells are ones which are found in a fully grown adult, and then they are restricted to certain niches in a particular organ. And the population is much less, so it's difficult identifying them as well as expanding them. So that's the bottleneck. But still, a lot of research has been going on adult stem cells as well to understand the biology behind what's happening in an organ. Yeah, coming to induced pluripotent stem cells. So when we go for autologous source of cells for treating a specific disease, we go for induced pluripotent stem cells because, as I said, adult stem cells, the numbers is much less, and again, the expansion is challenging. So now people, they have chosen induced pluripotent stem cells. So from the person who is suffering from a disease, the skin fibroblast can be taken, and this skin fibroblast, it can be cultured along with, or it can be transfected, I should say. It can be transfected with 4 factors, which is called as Yamanaka factor. Yamanaka, he is a Nobel laureate who received Nobel Prize in the year 2012 for identifying the or generating the induced pluripotent stem cells. So he's a Japanese basically. So he used 4 factors which I mean they are proteins: SOX2, c-MYC, KLF4, NANOG. And these 4 factors are transfected into a skin fibroblast. So when these cells are transfected with these 4 factors, they lose their identity as a skin fibroblast, which means they become undifferentiated. Now they become an undifferentiated cell. And now this undifferentiated cell can be differentiated into any cell of your choice by giving the proper medium, as well as the giving proper factors for them to differentiate into your cell of interest. So that's why we call these cells pluripotent cells. So that's what the image beautifully says. The skin fibroblasts, you reprogram them using the 4 factors. Then comes the iPSC. You can differentiate them. So this is being used for cellular therapies, disease modeling, as well as drug discovery. So extensive research has been going on in using iPSCs in all these 3 fields, as well as embryonic stem cells and adult stem cells. And again, I would like to emphasize one thing, that using adult stem cells is a little challenging than the other 2, but understanding the function and basic biology of an adult tissue resident stem cell is of utmost importance to learn the disease pathology, to learn what happens in the tissue with aging and other cellular processes that's been taking place in the tissue. So let's move on to stem cells in aging and disease pathogenesis. So why should we study these undifferentiated cells? So what's the need? Because they have a great role to play in aging as well as in disease pathogenesis. So these cells are the base. So when we talk about adult tissue resident stem cells, they are the base. So whenever there is at times of rescue, they are the ones that differentiate. They go maintain the tissue homeostasis by repairing or looking after what's happening at the time of disease pathogenesis or at the time of any mechanical damage. So they are the base. So understanding the association between stem cells and aging, as well as stem cells and disease pathogenesis, it is of utmost use in current medicine. So when talking about adult stem cells, neural stem cells, germline cells, and then the satellite cells are nothing else but the muscle stem cells. So they're all adult stem cells. They are all present in various parts of the body. For example, in eye, the limbal stem cells, trabecular meshwork stem cells, they're all adult stem cells. But with aging process, though they all come under the category of adult stem cells, they actually differ accordingly, like they differ in function as well as quantitatively. For example, if we take the hematopoietic stem cell, this is the blood, the hematopoietic system, the stem cells of the hematopoietic system, they quantitatively increase with age. But when we inspect the function, there's a clear loss of function. They're not as efficient as the younger stem cells that is functioning in the hematopoietic system. Now coming to the neural stem cells, satellite cells, the muscle stem cells, and the germline cells, with aging, they decrease. They don't increase like hematopoietic stem cells, as well as their potency or their function is also going down. So there's a clear loss of function. So there are many factors which leads to aging, just because the stem cell is also aging. So there are many factors that I have given here. Epigenetic regulations, there might be DNA damage or mutations with age, and the niche factors, the signals that they receive, and then their microenvironment, cellular senescence, and nutrient sensing, and the metabolism. So everything it affects, and it leads to stem cell aging, which in turn leads to deterioration of the system, may a blood system or a neural system, or it could be a muscle, anything. So this is with aging. And coming to the disease condition, so a clear reduction than the one which is seen in aging is observed when we talk about stem cells and diseases. So clear reduction than aging is seen in coronary artery disease, muscular dystrophies, and hypercholesterolemia. So in case of muscular dystrophies, like for an example, the stem cell pool is exhausted because muscular dystrophy involves wastage of muscles, and to repair the wastage, the stem cells are active continuously. It shouldn't be active continuously, so they're continuously active, and obviously the number of stem cells is going down. So this is because of the disease condition. In certain disease, the stem cell count goes down and it leads to a disease pathogenesis. In case of muscular dystrophy, the disease condition leads to a decrease in the stem cell content. So this depends upon the type of stem cell, type of adult stem cell. So this is what I would like to emphasize in this slide. And moving on to the main topic of the day, stem cells in ophthalmology. So adult ocular stem cells, I would like to talk on their location and their function. So this is one good illustration. So where the illustration, it shows the cornea, lens, the trabecular meshwork, retina, and just towards the right, the burn damage that happens to the cornea is shown, cataractous condition, glaucoma, and other retinal disorders is also shown. So where are the stem cells in cornea, lens, TM, and retina? So in these disease conditions or during the burn or mechanical damage, so how does these stem cells divide and how do they maintain tissue homeostasis? So let us look into what happens to these stem cells with aging as well as in disease condition in each and every ocular tissue. So let us start with the corneal surface, the ocular surface, the cornea. So I've just shown the image of a cornea. So here I'd like to talk about the corneal epithelium, one of the 5 layers. So the corneal epithelium, it sheds every 7 days, and it has to be replaced by another fresh set of cells. So where does these cells come from? It's obvious that there should be a stem cell to maintain the tissue homeostasis, to bring back the fresh set of epithelium. So where is this located? So this is another histology image I'd like to show. So this is the junction between the cornea as well as the limbus. So the stem cells for the cornea, they are present in the limbus. So very specifically, they are present in the basal limbal epithelium. This is where the stem cells are present. So whenever there is a need, a normal homeostasis where the epithelium is shed and there should be a replacement, the stem cells from here, they migrate and they divide, differentiate, they migrate centripetally towards the cornea, and then they replace the cells that are shed. So I think the next slide will, this will explain more clearly what happens to limbal epithelial stem cells. So I would also like to explain on the XYZ hypothesis. So you see the palisades of both the crypts. So the basal limbal epithelial stem cells, so they are present in the crypts, and these stem cells, whenever there is a need, so what they do is from here, they divide and then they move centripetally towards the cornea, and then they get fully differentiated into a corneal epithelium, thereby maintaining the homeostasis, replacing those cells that are shed. So here, when I say XYZ hypothesis, here X, it represents the proliferation and stratification of these stem cells. They have to first receive a signal, they'll have to divide, and then they'll have to find its way towards cornea. Y is the centripetal movement towards the cornea, migration, and Z, it's the desquamation and the superficial and the desquamation of the superficial layer. So to maintain the corneal mass, the X and Y, that is the proliferation of these stem cells and centripetal migration together, the sum together, it should make up the entire mass of the corneal epithelium. So this is the XYZ hypothesis. So in case of an LSCD patient, what happens here? Limbal stem cell deficient patient. The conjunctiva starts overgrowing the cornea and the cornea becomes opaque. So in that case, the limbal epithelium, a small explant, a 2 mm explant from the limbal epithelium of the healthy eye is always taken. Or if the damage or alkali burn or acid burn is being occurred in both the eyes. The buccal epithelial smear is another source where you can get the epithelium. This will be cultured in a good manufacturing process facility, and the cultured epithelium is taken back to the patients. And the success rate is much less in India. It was around 30 to 35%, and there were really good number of success cases, but the failure, the cases which didn't succeed were actually more. The reason behind is the microenvironment. So once when you transfer the graft onto the patient, the environment should support the graft. So when I say the environment should support the graft, always the limbal epithelial stem cells, they have supporting cells, or they have very supporting microenvironment. Just beneath the limbal epithelium, basal limbal epithelium, there are the stromal cells, stromal mesenchymal stem cells, which make up the microenvironment for the limbal epithelial stem cells. So if the degree of damage is much higher, and if the stromal mesenchymal stem cells are also affected, what happens is whatever graft is being taken to the patient will not be accepted. So this was the main reason why there were more cases which couldn't be succeeded when the graft is being taken to the patient. So there is a separate population of stem cells called stromal mesenchymal stem cells in the anterior stroma, which supports the limbal epithelial stem cells. So it's the microenvironment for the limbal epithelial stem cells. So in my laboratory, so all these conditions in an LSCD patient, they have recorded the in vivo confocal microscopic, using in vivo confocal microscopy, they have recorded the image in normals as well as in LSCD patient, and the degree of damage in an LSCD patient, whether it is superficial, the damage is superficial, or even the stroma is affected. And just because the stroma is affected, the one that you the one that you graft onto the patient becomes a failure. So everything is being recorded, and there are a couple of publications which I have attached along with the presentation. So next, moving on to trabecular meshwork stem cells. So this is my area. I work on trabecular meshwork stem cells. So this is a full illustration on the normal functioning of the outflow pathway. So where are the stem cells in the outflow pathway? Yes, there are stem cells. They are present in the, let me go to the next slide. So this is my my own histology image. So the stem cells for the trabecular meshwork, they're present in the non-filtering region of the TM, or it's also called the insert region just because it inserts beneath the corneal endothelium. The region is also called anterior non-filtering region, Schwab's line region, and insert region. So there are many terms that can be synonymously used. And just towards your right, I have another image. So this shows exclusively the Schwab's line region. Between the arrows, there are a stretch of cells. They are said to be the stem cells. The homeostatic property of these cells were experimented using human organ cultures. So laser trabeculoplasty is one surgical method to treat glaucoma. So when the organ cultures were treated with laser, the researchers observed that the cell, due to organ laser trabeculoplasty, there will be burns in the filtering region of the TM. So to repair the burns of the filtering region, the cells from the non-filtering region, they migrated towards the filtering region, which means they had a homeostatic activity, they had a proliferative potential to migrate towards the filtering region just to repair the laser burns. So this is a property of a stem cell. And then looking into the cellular morphology, the ones in the non-filtering region, they are more cuboidal, and then the ones in the filtering region, they are obviously different from the ones in the Schwab's line region. So there are morphological differences, and function-wise, they were able to maintain homeostasis by repairing the burns. So this was the first study where they show the homeostatic potential of the trabecular meshwork stem cells. Yeah, this is also a very own image of mine. So I tried locating these stem cells. There were a couple of studies where they've located, but here I have used two markers. So the green panel, it represents a marker which is called ABCG2. It's a transport protein, and P75 is a neural crust derived stem cell marker. It's a TM is neural crust in developmental origin, so that's why I've used a lineage specific stem cell marker. And the third panel, it shows the nuclear stain. The blue, the ones in the blue, they are the nuclear stain. And it shows the merged image of both the green and the red images. So the green is nothing else but the expression of ABCG2. So where the ABCG2 expression is, it is there in the non-filtering region. That's what I've marked here. And P75 is also seen in the non-filtering region. And just beneath the images, you can see the non-filtering region in a higher magnification. You can see the cuboidal cells, which expresses the stem cell markers. So this is my favorite image, and this has been published in the journal Eye & Vision. And upon quantifying these stem cells, what I found with aging is there was reduction. So I had 3 age groups: below 30, young age, 30 to 60, middle age, and above 60, the older age group. So with aging, when I counted the ABCG2 and P75 cell count, I was able to achieve a reduction. So just like satellite cells, just like germline stem cells, there is a reduction. So I did the same in glaucoma using glaucomatous donor eyes. So I found a reduction in the stem cell content higher to age-matched controls. So there were many glaucomatous donors of age 80. So many questions that rise to me was, with aging, I have a stem cell loss. So in glaucomatous condition, whether the loss is due to stem cells or is it due to disease pathogenesis. So it was clearly due to aging as well as disease pathogenesis, because I strongly give this statement because the reduction in the stem cell content higher than the age matched controls. When I say I had donor eyes above age 80, I had a set of control normal eyes also above the age of 80, and I compared the stem cell content. So this is my very own work on trabecular meshwork stem cells. And moving to the transplantation. So when we give an external stem cell therapy, what happens in a glaucomatous condition? So there are studies where they use trabecular meshwork stem cells. They isolate it from the humans, they expand it. There are some studies where they use iPSCs and then they differentiate that into TM. And that's been transplanted into glaucomatous mode. Intracameral injection of these stem cells or stem cell derived TM cells. Then they're injected intracamerally into a mouse model. So it could either be a glaucoma model or ocular hypertension model, high IOP model. These are the following results. The TM cellularity was improved. So usually in a glaucomatous condition, the TM cell loss is much higher. The intraocular pressure was reduced upon stem cell transplantation. The aqueous humor facility was enhanced. The ECM modification, because in glaucomatous condition, ECM remodeling is another result of glaucoma. ECM modification has happened. So this was one recent study where they talk about the ECM and myocillin secretion. So in a mutant condition, the myocillin is being withheld within the cell, and in a normal condition secreted out in the aqueous humor. So analysis of the aqueous humor after the stem cell transplantation found the secretion of myocillin, which means that yes, the homeostasis or the normal functioning is possible after a stem cell transplantation. The cells has become healthy. So I have just given a cartoon of where the cells have become healthy. But there was one disadvantage, a drawback. The endoplasmic reticulum in these cells, in the cells where it persisted. So they were high stress because endoplasmic reticulum, it is the protein machinery of the cell. So there are still issues regarding the endoplasmic reticulum. So that was one drawback where all studies have pointed out till date. Yeah, move to the lens epithelial stem cells. Yes, there are stem cells in the lens as well to maintain the homeostasis, because lens fibers are found even at the age of 87, which means there are stem cells somewhere which is actually producing these lens fibers even at the age of 87. So this is, I have given the image of lens here, and then I have divided the lens into 4 zones. So this is based on a work that has been going on in my lab, and my colleague Saranya, she is taking up the work on lens epithelial stem cells. So we have divided the lens into central zone, germinative, transitional, and equatorial. So the lens anterior epithelium is divided in such a way. This is all based on the literature. and we're trying to characterize it. So we're characterizing it currently, but there were previous studies and there was so much of controversies. So there were people where they claim that the central zones consist of the stem cells, and there was one group debating on the presence of stem cells in the equatorial zone. So where are the stem cells present? We do not know. So we are trying to look into the location, and my colleague, she has identified the stem cells in the central zone of the anterior lens epithelium. So I have also attached the title of her abstract here. So this is an RO abstract which was supposed to be presented this year at RO Baltimore, and still I think you can view her abstract in the RO website, and beautiful work is being carried out from our stem cell team from AMRF. So you can just look into. And very importantly, there were many studies on animals, but there was one study, Lin et al. 2016. It's a Nature publication. So in this publication, the person claims the central region harbor the stem cells, just like what we are talking about right now. So when cataract surgery was carried out on the pediatric patients, so using a minimally invasive technique, the central region, the hole was not made in the central region. The hole to remove the lens fibers was made at the periphery, leaving the central region intact. So after 2 weeks, they recorded the formation of lens-like structure right from second week, and they recorded it till 4 months. And at the end of 4 months, they were able to see a complete lens-like structure in the human. But still, the arrangement of fibers was another question. But the arrangement of fibers as well as the vision was normal when it was carried out. The experiments were carried out on animal models like rabbits as well as on monkeys. So leaving the central zone unaffected, he claims that a whole lens-like structure can be regenerated. So this is one, but this is really one great lead to say the central zone harbors the stem cells. But we need to know more on it. Really, whether the central zone harbors a stem cell, what kind of properties do they have, and what happens with cataract, and do they have something in the formation of cataract? So really, a lot has to be done still on the pathogenesis of cataract. And now moving on to the retinal stem cells, more light has to be shown on the retinal stem cell area. So today I'll be mainly concentrating on 3 layers of the retina. One is the ganglion cell layer, the next one is the photoreceptor layer, and retinal pigment epithelial layer. So the native stem cells, like as I said in limbus and TM and lens, I couldn't get more literature on the native location of the native stem cells. So that's what I told. A lot of research has to be done on the endogenous stem cells. Still, the location is a big debate, and we do not know where the stem cells of retina is located. So we'll just move on to the 3 layers that I have mentioned one by one. So retinal ganglion cells. When we talk about optic neuropathies or any neurodegenerative disease, retinal ganglion cells is going to be the pathological site. So they are involved in neurodegeneration. RGC death is going to be the major RGC cells. RGCs are going to be affected predominantly. So there are no endogenous stem cells to rescue. That's why we experience an RGC cell death. So this is what many studies they claim. Because whenever there's going to be a cell death, there should be a stem cell that should come into play to rescue the condition. But in retina, no such stem cells and no endogenous stem cell activity is seen. So people, they thought they can go for some external sources. So what are all sources did they use to look into or to try to reverse the pathological condition? So they used human fetal retinal progenitors. So retinal progenitors taken from the human fetus, the mesenchymal stem cells, and bone marrow derived stem cells. And very importantly, they use the Muller glial cells. Sometimes they say that the Muller glia, which is intrinsically present in the retina, can also program itself towards RGC lineage. So this is one study, and people also claim Muller glia might have the stem-like properties, and then they might help in RGC neuroprotection. So this is one. So I use the word might. So we do not know. There are more research which has to be carried out in this area. So all these three types of stem cells, they are being transplanted into the retina. So human fetal progenitors, they were transplanted into the vitreous, and this is a clinical trial that is ongoing on humans. And on rodent glaucomatous models, it actually showed neuroprotection. And even in other retinal diseases, the bone marrow derived was not done on a glaucomatous model, it was done for other retinal diseases. So as a baseline, I can say neuroprotection. Neuroprotection was observed in one clinical trial and in other retinal diseases where they have transplanted the stem cell. So there were many studies. So I just tried narrowing down. So I don't want to crowd a lot in my slide or dump a lot into your head. So this is on RGCs. And next, coming to the photoreceptors. So they are the light responsive cells. They are terminally differentiated. The term terminally differentiated, it means just like RGCs, they don't divide. So once they're developed, they don't divide. So they are the pathological site for many diseases. AMD, age-related macular degeneration, retinitis pigmentosa, diabetic retinopathy. So yes, stem cell therapy has to be applied to such cells as well to rescue all these disease conditions. So there was one study, so I'm just picking up one good example. There was one study where raw photoreceptor precursors, so these cells were derived from stem cells and they were injected into the mouse retinal models. So when they were injected, they were able to see they were able to get good results. So they were able to see that the vision was restoring in the mouse models. And how did they interact? So when you inject stem cells, so how do they interact with the right cell, or how do they communicate with the recipient cell? The one that we inject is the stem cell, they are the donor cells, and the one that receives or the one that is trying to get help is the recipient. So people they identified a or it could be microRNA from the transplanted stem cells. So they were transferred into the photoreceptors. So in such a way that they received help from these donor cells and they were trying to restore vision in the animal models. And now coming to retinal pigmentum. So again, AMD, Stargardt's, other macular disorders. So it involves retinal pigment epithelium. So here, people have they have used both embryonic stem cells, they have used induced pluripotent stem cells, and then out of these stem cells, they tried differentiating RGC progeny. And then the results were actually amazing. Like they were able to rescue vision in these models. And when I was talking about native stem cells, again, even in the RPE, retinal pigment epithelium, we do not know where the native stem cells are present in the eye. But in culture, we were able to see the RPEs. They had stem-like property, or they express stem cell markers. So there was one study which shows that cultured RPE, they harbor stem-like property. So this was a study from the Neural Stem Cell Institute from New York. So by Sally Temple and her group. So this was one study where they identified by culture that yeah, they have stem-like properties, but still in native, it is something that it's unknown. So even in our group in AMRF, we are trying to look for the native stem cells, but still there are a lot of challenges when we talk about retinal pigment epithelium. So it's all due to the pigments, the melanin pigments, the lipofuscin, identification, or any kind of method like immunostaining. So it's actually a problem due to the pigments. So we are trying to resolve the issue and then try identifying them, identify the location in native. And yeah, coming to the therapy. So coming from the right from the cornea, so there are many preclinical and clinical trials that are going on. So corneal endothelial regrowth is one thing that is going on, and the stromal scarring or the fibrosis in the stroma. So this can be reversed by using the stromal stem cells. So this is being carried out in India by L.V. Prasad. And cultured limbal epithelial cells, which I was earlier talking about when I was talking about the limbus. So that's already there in the clinic. And when it comes to lens, as I said, in pediatric cases, they were able to look for the generation of whole lens-like structure, but still there are many questions to be answered. And in TM, so the TM, they're trying to replace the glaucomatous TM cell, affected cell, by a normal cell in such a way that it restores the intraocular pressure. So the homeostasis of the entire T restored. So we are trying to do that in the field of TM and retina. So whatever I spoke on retina is given here. Again, they are using induced pluripotent stem cells, embryonic stem cells, and there are much studies going on only on animal models because patient safety is a major concern. A lot of studies has to go through the preclinical trials and clinical trials before it's being taken into the patient. So there are more animal model studies when it comes to retina. And this is the clinical pipeline for the cell-based therapies that's going on in eye diseases. And we'll have to, I think we'll have to read a lot, or we'll have to work a lot on the basic mechanisms of stem cells. So what is their actual functioning in the tissue homeostasis, where they are located, and if we gain all these knowledge, I think we will be able to design or develop very cell-based therapy for patients who have been suffering with so many dystrophies, maybe corneal, or in case of lens, TM, as well as in retina. And when we talk about therapy, like how do they actually generate these and then inject them into a patient? So this is the next slide that I'm going to talk about. Bioengineering. So it could either be iPSCs, it could either be induced pluripotent stem cells or tissue-specific stem cells. All these cells will be expanded. So based upon the condition, they will select the cell. This will be expanded, and this expansion is being carried out using the hydrogels, bioreactors, nanofiber matrices, and everything. And then they'll allow the cells to differentiate into that particular lineage. And what they do is they do a selection, nothing else but isolation and purification. So this is a very important step where you will be picking only the cells of your interest. So a differentiated cell of your interest, and other cells which is not coming under the category will be eliminated in this particular step. You'll be taking only the pure population of differentiated cells that is to be taken for transplantation. And before it goes for transplantation, either it has to be embedded on a matrix, and then it has biomatrix, and then it has to be transplanted. So that's what is shown here, cell and tissue assembly. So always you need a matrix or an assembly so that you will have the cells on it, and then you transplant. Or directly, in certain cases, directly the stem cells can be transplanted. And either the matrix is now grafted, or the cells, they are transplanted directly into human for treating the particular disease. So this is the bioengineering. This is how it happens. And this is how the tissue banking and cell banking, and as well as the companies that generate cells, differentiated cells from the iPSCs or embryonic pluripotent stem cells, or from the tissue-resident stem cells. This is how they work. And yes, there are major concerns. So challenges in bringing ocular regenerative therapy to patients. So certain points I have mentioned then and there. One major issue is going to be the immune response. So this can be, this issue can be addressed by, alleviated by the HLA typing. So there are certain companies, they are trying to match the HLA between the donor and the recipient in case of allogenic transplants. So this is one issue. HLA matching has to be done so the graft can be easily accepted by the recipient. And next, I was talking about companies or laboratories where they expand these stem cells or they differentiate these cells. So this expansion and differentiation of these stem cells, it has to be repeated more than once. So to check the authenticity of the results, like how many ever times you repeat, you should get the same results. So the results should not be deviated. So that's the main concern. So if the results are going to be deviated, that's going to really create a problem upon transplantation. So you'll have to go for many trials, or you'll have to repeat it many times before you take it to the patient. And yeah, when we talk about stem cells, when we talk about the cells that are differentiated from the stem cells, like I think there are other characteristics that we still need to understand. So using the current characteristic of a stem cell, I don't think the current characteristics is adequate. So I have mentioned few here. So identifying them based on the markers, purity, their potency to differentiate into a specific cell type. So I think these factors are inadequate. More characterization can be done so that the transplant that you do, either the differentiated cell from a stem cell or the direct stem cell transplantation that you do, will be a success. So to make it a success, more knowledge or more characterization is essential. And as I said before, animal models, they are the main here. So they're the main players. Like whatever we try discovering, it's been treated or it's been tested using animal models. So only after passing these tests it comes to the clinic. But even we use the animal models are problematic. So always the results out of these animal models, they don't come out as we predict. For example, glaucoma. So when we take glaucoma, it's a multifactorial disease, and then there are a lot of genes involved. There are a lot of genes involved in the pathogenesis, a lot of mutations involved. But when we talk about animal models, there are only few animal models. They don't cover all these genes or all the external factors that causes the disease. So one major or one predominant model that is being used in glaucoma is a mouse model. It's a glaucomatous mouse model. So that model is transgenic Y437H, which means this mice is being mutated in myocillin gene, and this myocillin, in the myocillin gene, the tyrosine amino acid in the 437th position is being replaced by histidine. By making just a single base pair mutation, they have made the mouse glaucomatous. So this is just one model. So there are other genes involved. So what happens when you transplant these stem cells to a patient who has other kinds of mutation? You have checked it only in a one animal model. So we have not looked for the other factors that are involved. So they say yes, they are problematic again. And teratogenic. When it comes to iPSCs, the teratogenic properties are much high. That has to be clearly eliminated before it goes for transplantation. That cancer forming efficiency has to be tested again and again. It has to be repeated before it goes for transplant, or it goes for teratoma formation after transplantation. So that is another issue when it comes for taking therapy to the patients. And this is actually a tragic reminder. There is one study where they have interpreted on this particular result. I have given an example here. So intraocular transplantation of liposuction fat derived cells. They have blinded several patients because they have sidestepped certain challenges that I have mentioned currently. Yes, they had tragic results coming out. So to eliminate this, so to put patients' safety first, I think researchers as well as clinicians, we should work hard to see we kind of eliminate all these challenges to make better results in the field of stem cell biology as well as in the transplantation biology, regenerative medicine. And yeah, these are the references used in my presentation. Yeah, thank you.
Great. Thanks a lot, Suganya, because that was very enlightening to know so much work has been done in stem cells alone. Because stem cell is one area which most of the ophthalmologists, I think even doctors for that matter, they're still not aware about the potential of stem cells and how we can easily translate the stem cells into therapy as well. So I think she has covered everything beautifully. So I just want to summarize whatever Yogapriya has mentioned. So let me just share the screen for a while.
Yeah.
Okay. To quickly summarize whatever she has mentioned, first and foremost is stem cells are primitive or undifferentiated cells, and they can undergo these 3 Ds. They can be dormant, they can duplicate, or they can differentiate. So it's like a monkey. So just imagine a monkey is there, a monkey is staying quiet like a dormant stage. The monkey is going to reproduce to form another monkey. There is the duplication part of it. Or the monkey can evolve into a man, kind of a differentiation. I don't know. It's not a scientific way to compare, but to make things easier, the 3 Ds are going to characterize stem cells, right? Okay. Next is going to be, what is the
That was perfectly put.
Right. Thank you. So that is
that was perfectly put.
Yeah. So the ultimate aim of stem cell is to maintain homeostasis. For that matter, in our body, there is a disease state, there is a healthy state. So it's all a battle between a disease and between healthy state. So stem cells are going to try to maintain that state of healthy balance. And we have 3 types of stem cells, as Yogapriya mentioned. We have an embryonic stem cell, the induced pluripotent stem cells, and adult stem cells. The ESC and iPSC, they can transform into any cells. The one difference with the embryonic stem cell is that they can also form an extra embryonic cell. That is what we call as a totipotency. Whereas an induced pluripotent stem cell can form into any cell, but not an extra embryonic like placenta. So they're called as a pluripotent cell. The ASC or adult stem cells are going to form only a particular lineage of cell. They can't transfer into any cell for that matter because they're committed. They're committed towards one lineage, so they can just form only that particular type of cell. For example, hematopoietic stem cell forming an RBC. And she mentioned a very interesting word called Yamanaka factors. They're nothing but these proteins were added to the skin fibroblast so that they can be converted into any cell. Is that right, Yogapriya? Is that the way? Yamanaka factors?
Yeah. You transfect those factors into a fibroblast. Okay. In such a way, the fibroblast loses its identity and it becomes undifferentiated. Now it's a stem cell. Right. So now you can coax that stem cell towards any lineage of your interest.
Okay, that's great. Good. So next is the most important concept to understand as far as stem cells are concerned is note that aging and disease can cause a reduction in stem cells. But the intensity of loss is going to be more in case of a disease or in case of a pathology. Like she mentioned, trabecular meshwork stem cells were found to be reduced in aging as well as in a patient with a glaucoma. So that is a very important thing to understand. Aging and disease are 2 processes which can cause reduction in stem cells. As far as eye is concerned, there are multiple stem cells yet to be explored, for example, in conjunctiva or maybe in sclera. But the most commonly studied ones are C for cornea, lens, trabecular meshwork, and retina. As far as cornea is concerned, it is going to be the anterior stroma and the basal limbal epithelium. These 2 are the common areas where you can find the stem cells as far as cornea is concerned. The more important is going to be the limbal stem cells present in the basal limbal epithelium. These limbal stem cells are going to replace the corneal epithelium by migration. Okay. And after bone marrow transplant, the most widely used stem cell therapy is going to be limbal stem cell transplant. Now, she also mentioned about the XYZ hypothesis pertaining to the limbal stem cell migration. So what is X? X is proliferation of stem cells. Y is the migration of those stem cells to replace the epithelium. Z is the loss of pre-existing epithelium. So always it should be the X plus Y should be equal to Z. The formation should replace the loss. That is the concept of XYZ hypothesis. Coming back, there is lens. So as far as lens is concerned, the epithelium of lens is going to have these stem cells. T, trabecular meshwork. To be more precise, it is a non-filtering trabecular meshwork which is going to have the trabecular meshwork stem cells. And these trabecular meshwork stem cells can form or can convert into 2 types. One is the trabecular meshwork cells by themselves and also the endothelium of the cornea. And the
I think I just missed that point, I guess. Yeah, I didn't mention it on the endothelium. Yeah. Okay. So they are multipotent. So these stem cells present in the non-filtering region. So they are in the junction between the TM and the endothelium.
Right.
So it's very interesting to know that These are the pet cells, progenitors of endothelium and TM. But the mechanism by which they become an endothelial cell or by which they become a TM cell is still unknown. So what are all the molecular factors? What are all the proteins which make these cells into an endothelium or a TM? So I think this is a very interesting question to be asked, and very interesting question to be answered by any researcher by doing research on the molecular cues that makes these stem cells differentiate into two different, because TM morphologically they're different. Endothelial cells, they are hexagonal. They have different functions, but they're all neural crest in origin.
Exactly.
Developmental origin is going to be neural crest.
Great. So just understand the importance of embryology here. Since neural crest cells is going to give both trabecular meshwork as well as your corneal endothelium. So that is the implication of the embryology here. So the trabecular meshwork stem cell can convert into a trabecular meshwork cell, also an endothelium of cornea, right? Okay. So let us go to the next thing. Let me just quickly reshare the screen. Right. The next thing what she mentioned was about the biomarkers. See, the stem cells are going to express a proteins or antigens. Those particular specific proteins are called as biomarkers. So we are going to attach an antibody to the protein or to the antigen. So it's going to be an antigen antibody reaction so that we can locate the particular stem cell. So she mentioned specifically two important biomarkers, I think. That is going to be the ABCG2 and P75. So these two proteins are more expressed in stem cells. So when you can...
Yeah, can I make a small... can I just interrupt here?
Yeah, please.
Yeah, so every stem cell, like for example, Limbus or TM, they have their own set of biomarkers. So ABCG2 and P75, they are specific for TM. So when we talk about Limbal, it's ABCG2 and P63.
Great. So for
when we talk about lens, yeah.
So it is going to be ABCG2, the same ABCG2 and P.
Yeah, and P63.
P63 for Limbus. Okay.
And very importantly, ABCG2 expression should be higher. So high expression of ABCG2 in both the cases, and P75 for TM and P63 for Limbus. And when it comes to lens, it is SOX2. So every adult stem cell in the eye, they have different markers based on which we can identify them. So it's not the same for all stem cells. So there are various properties. Limbus, it's an epithelial cell. And when it comes to TM, it's a neural crest or a mesenchymal stem cell. Lens, it's again epithelial. Right. And when we come to retina, it's of a different lineage. So they all have different properties and different markers based on which you can identify.
Right. Good. Great. So those are about the biomarkers. Now coming to how do we actually use the stem cells in therapy? So simply put, when I find a stem cell, I'm going to isolate that stem cell, purify the stem cell, culture them or to make them grow, and I add those stem cells to a scaffold or to a carrier, and I'm going to put it or inject it into the required area. So this is going to be the sequence by which we make use of the stem cells available, right? Or even without using a scaffold, we can even directly inject those stem cells into the required area also. She also mentioned about the retinal stem cells. Retinal stem cell, because of the complex organization, it's very difficult to isolate that stem cell, and it is also very much an unexplored area. So it is also a great area of research as far as retinal stem cells are concerned. And last important thing I want to tell is it is an evolving concept. It's like this corona currently. Nobody knows how is it going. We are not even sure how this is going to transform. But let's hope that all positive is going to happen as far as stem cells are concerned. It is a potential area of research, and we hope lots of untreatable diseases can be cured. So just have a couple of questions, but I just want to tell the same thing. To re-emphasize the last slide what Yogapriya was using, if you can just focus on this, in this slide, you can see there are multiple cornea, lens, trabecular meshwork, retina. Most of them are in preclinical or in phase 1. They are yet to be used on the clinical side. So probably the only area which is clinically used is going to be the limbal stem cell deficiency.
Limbus, so far, yeah.
So we're going to use this limbal epithelial cell. So the most clinically relevant area of stem cells in ophthalmology is going to be limbal stem cells. So that is why that question is being asked in the exams for the ophthalmology residents as well. Now, I just want to end with a small table. This is one of the table I got from a review article on stem cells in ophthalmology. This table kind of brings everything to one circle. It kind of shows how we actually apply stem cells in treatment for a particular ocular disease. Just look at this once. Now, in this table, they're going to talk about ongoing or recently completed stem cell based therapies in retinal disease. That is the title of the table. So they're mentioning 4 important parameters. One is, what is the type of stem cell? What type of stem cell are we using? Is it a bone marrow derived stem cell, which is the adult stem cell, right? Is it a bone marrow derived stem cell, or is it an iPSC or induced pluripotent stem cell, or is it going to be a human embryonic stem cell? So all 3 stem cells are being used. What is the mode of transplantation, mode of injection? Is it intravitreal? Is it subretinal? You can see one area where they have a retrobulbar subtenons also they are being used. And look at the diseases. Atrophic age-related macular degeneration, ARMD, is one area where still a lot of research is going on as far as treatment is concerned. Stargardt's disease is another disease like Yogapriya has mentioned. And what is the study phase? Where are they using? So just understand, this is how they actually make use of stem cells in ophthalmology and the treatment of ocular diseases. Just got some couple of questions for Yogapriya. Now, can stem cells undergo a tumorous growth? Can they become cancerous?
So it's based on the mutations. So if they acquire mutation and if the mutation leads to uncontrolled cell growth, yes, they can cause cancer. But I think if I start talking on this, I'll talk more on cancer stem cells. So which is actually a different topic. But to be very precise to your question, yes, it's based on the mutations they acquire. So with aging, because as they infrequently divide, and with aging, this frequency might change. And due to the change in this frequency of divisions, there might be certain genetic mutations that they acquire. Because of those mutations, yes, disease pathogenesis takes place, and tumorous growth, yes, it is possible. But I think we'll have to study even more on that as well, because yes, as you said, stem cell is a growing field. There are a lot of questions yes to be answered to understand like what is happening in a human body.
Correct.
And being stem cells being the base of a specific lineage, yes, we should know a lot about them.
Right. The second question is, you already discussed about it, but I just want to clarify that. I think death is one important process in any pathology for that matter. Be it in aging or a pathological process, eventual is going to be death, either by apoptosis or by necrosis. So what a common man will ask is, can we reverse aging with the help of stem cells? Is it a possibility?
You can slow down. You can slow down the process of aging. But still, when we talk about stem cells, we cannot give a clear cut and a very strong statements always. We can't make those statements, right? Because we can use stem cell therapies, but still you can only slow down. And moreover, there are other genetic factors in the human body that is associated with the aging and the disease pathology. So, yeah.
Yeah, okay, good. The second question is more pertinent to the ophthalmologist. See, when I see a patient with a glaucomatous optic atrophy, like a 0.9 cupping, complete pallor of the disc, the total optic nerve that is atrophied, I'm going to tell him or her that, see, that is the visual prognosis for you. You can't have any more better vision. So I'm going to give a very grave visual prognosis for that patient. So with the help of stem cells, can we reverse this, or can we replace the dead cells with the living cells? Is there a scope of hope for them to retain their vision?
Yeah, if you had seen my the therapy slide, so where I have also mentioned near TM, the glaucomatous TM cells, the affected ones, we are trying to replace it with a normal TM so that it maintains the tissue homeostasis. So that's ongoing. And to add more on this, yes, studies on animal models, they show promising results, but none has been tried out on humans. As I said, we'll have to overcome a lot of hurdles to touch patients.
Correct.
Yeah. So The animal model may not revert 100% back to normal, but definitely the condition is going to get better. The tissue homeostasis is going to act better on a stem cell therapy.
Right.
So, yeah.
Okay. So still it's ongoing process. We're not sure it's going to be yes or no, but currently, yes, there is some kind of hope based on animal models. But how far it's going to be translated to humans is still a question mark, right?
Yeah. Good. So we need more studies on it, more on the biology, more on the mechanisms.
Good.
Yeah.
So that's with the summary of the talk and with the question discussion as well. I know it's a very dry area, but it's very exciting when you really understand and when you hear from a very young talent and a budding scientist like her, to really know what is the scope of stem cells is. So what we'll do is we'll try to maximize the utility of this lecture. So what we'll do is we'll try to create a PDF. So what we'll do is we'll try to create a PDF with all the points, whatever Yogapriya has mentioned, the entire summary or the gist of the entire presentation, along with flowcharts and pictures. And we will add a very important note on limbal stem cell deficiency, because 2 questions are being asked as far as residents are concerned: the role of stem cells in ophthalmology. The second is going to be limbal stem cell deficiency. So knowing about these 2 questions and the answers for them is very, very important. And not just that, for those who really want to take it above their exams, we'll try to attach some review articles, some ongoing research methodologies, like all the exciting updates for postgraduates. And if you're really interested, you can even take it up further. I think that ends with this discussion on stem cells. So thank you so much, Yogapriya. I know it's a very,
thank you so much for having me here. And I would like to add some more here.
Yeah, please.
Yeah. So research in stem cells. So it doesn't end only with the researchers. We definitely need help of clinicians, because with your knowledge on the disease pathogenesis, because you address patients day to day, and because we identify stem cell in a particular location, to dissect that particular the stem cells from that particular location, we need your expertise. And not just that, even now in glaucoma, I just give a very plain statement saying stem cells go down in glaucoma. But what happens in an early glaucoma? What happens in a late glaucoma? What kind of therapy you're giving? What to treat the glaucomatous condition, the topical drugs, or the surgery that you do, how it affects a stem cell? I think more of clinical input has to be there to address a lot of such questions. Because whenever I presented the data on glaucomatous TM, trabecular meshwork and stem cells, they asked me whether the donor has had a history of the duration of glaucoma, the medications that they were using, and then the surgery that they undergone, whether the medications that they used had an effect on these stem cells. So there are a lot of questions which we would like to have your help. So we researchers together with young ophthalmologists, I think we can do wonders in the field of stem cells, because your knowledge and our technical skills will actually bring in better results, and we can design better therapies for patients with the disease pathology. So this is what I wanted to finally convey. We need the help of clinicians in many areas. We need your input.
Great. So let's work together. That's what you want to say.
Yeah, let's work together.
Great. So thanks a lot, Yogapriya, because that was a very enlightening talk. Because so far I knew the only type of stem cell is limbal stem cell. I thought that is the only stem cell available in the eye. But after listening to your talk, I knew that so much is there, so many areas, niche areas where we can still explore and make use of it in our clinical practice. So that's it for today's edition of TalentsTeach. So thank you so much. Hope you enjoyed this talk. Thanks a lot. Bye-bye.